Okay, well, thanks, Dennis, for the intro. You kind of took care of my second slide for me there. But I'm excited to be here to talk to everybody. This talk was really great, or putting this talk together was really nice because it forced me to put my research in a context that would appeal to the masses, because sediments really aren't usually that exciting to people. So I hope I've come up with something a little more exciting for you guys today. Okay. So as Dennis said, Ph.D. from Georgia Tech. Then I was over in Sarasota at Mote Marine Lab. Worked a lot on red tide and harmful algae blooms over there, as well as some connections to sediments. I moved over here in 2018, January, so a little over a year ago. It's a great place to work, great colleagues, great science, but also much better waves at Sebastian Inlet. And we also have a hell of a football coach in Lane Kiffin, and he's extremely entertaining. So as he said, I direct the Geochemical, or the Geochemistry and Geochemical Sensing Lab. And I have an affiliate appointment in the Department of Ocean Engineering and Geosciences as well. And currently our lab is all of myself and one student, Andrew, who's back there. Thanks for coming, Andrew, although you should be in the lab. That is our lab down there, by the way. It's cool. It's a lot of fun to set up a new lab. It's like being a kid in a toy store or candy store. So what do I do? It's a lot of different things. It depends on the day. We can be a sediment biogeochemist, an aqueous geochemist, chemical oceanographer, environmental science. Overall, it's really, our lab is just a nice mix of fundamental research, technology development, and applied research. So chemical oceanography, what is that? In one slide, it's just looking at the reservoirs of various chemicals in the ocean. So that's basically it, and looking at their size and their residence time and fluxes as well. So for instance, here is, we're looking at iron. We just took a slice of the ocean from the North Atlantic to the South Atlantic. And you can see, for instance, near the equator, you have the Mid-Atlantic Ridge, and you have underwater volcanoes that are spewing out some iron. So this is sort of the most common picture you'll see in oceanography. We also developed new instrumentation to collect this data at higher time in space frequencies. Now, when I put my other hat on as a marine biogeochemist, we're more looking directly at mechanisms and processes. So it's not so much where the elements are, it's more how they're transferred, the processes, the reactions, thermodynamics, kinetics, things like that associated with the transfer between the various reservoirs. Okay, so like I said, this is forcing me to put my work in a new context, and it's a new big idea. When I say that, we all went to our retreat as faculty last September, and each of us presented our crazy big idea. And this is mine, and I hope you guys like it. I hope other people like it. I think it's pretty cool. I've gotten pretty good feedback about it. What I talk about today is going to be a range of simple stuff to difficult stuff, but I hope I at least get you walking out of here thinking more about sediments than you ever have. So I'm going to talk about soils, though, obviously, and why. There's a little backstory. I would go out and make all these measurements in marine sediments, and I would see that specifically iron hydroxides in sediments would contribute an element of resilience. It would make these sediments more healthy. It would protect the ecosystem services that these sediments would provide. So sediments could essentially be considered a resource. The sediments, these resources are being threatened from local global eutrophication, which I'll talk about, climate change. So rapid conservation is needed. And considering how closely related soils and sediments are, we may as well learn from soil conservation, because we've made some serious mistakes in the past, primarily being a lack of ownership. People didn't really care about the soil, led to some unhealthy practices, some selfish practices, overall less resilience of the soil system, and less ecosystem service output. And so there's a lot of questions remaining for sediments that we already kind of know the answer to for soil. I mean, have you ever heard of sediment conservation, like sediment geochemical conservation before? I haven't. If you Google it, nothing comes up. So I think we're on to something. Okay. So a little rewind. Who remembers learning about the Dust Bowl in high school? Cool. Pretty much everybody. Who, since we're in Vero, who remembers the Dust Bowl? Come on, that's a joke. No hands went up. I was hoping somebody would raise their hand. But anyways, so the Dust Bowl was ecological catastrophe. It started as early as the 1860s, as when things really happened. Everybody moved west. The government offered very cheap loans to folks for land, and they said, move out west. And so everybody moved out west, and the only catch was they had to live on the land, I think, for 5 or 7 years and develop the land some. And unfortunately, we didn't really understand that much about soil science back then. People actually thought that if you moved out there and started tilling the soil, that the climate would change, the rains would follow. They also thought that soil couldn't be exhausted. It was one resource that was infinite on the planet. So there was the great plow up in World War I. Grain prices really rose, and 2 times the land area of New Jersey was plowed in just 5 years. There was new technology as well. The lister right here, which you can see is sort of vaporizing the soil layer, replaced the lister, and this is sort of a new age lister, but you may have seen pictures of a lister with being carried by a mule. A mule is pulling like an under the soil wedge, and you're not really vaporizing the soils, you're just sort of turning them over, so it's much more gentle on the soils. There was, in 1929, obviously the Great Depression, and the wheat prices dropped by a factor of 5, so what did people do? They just tore up more land to make a bumper crop to try and make up a difference and turn a profit. And so they actually moved further out west into no man's land where it's really, really desert lands. It's very difficult to grow things, especially wheat. So one of the most tragic things, especially if you like animals a lot, is the jackrabbit drives that happened. The biodiversity was so destroyed just by people destroying the soil and planting one single crop that you ended up with a jackrabbit infestation. People would have to actually pile these animals into a pit and then beat them with clubs. It happened. So overall, we had decreased ecological health and resilience. The topsoil turned to dust. You had no more sod or windbreaks, no buffalo grass, which really acted as a staple, sort of holding the soil in place. There was no food for cattle. Then you combine this with the worst drought in recent memory. It lasted for 10 years. It brought these gigantic black blizzards, 10,000 feet high, these dust storms you couldn't see your own hand in some cases. 1935, Black Sunday happened, destroyed the entire crops of several states, and folks in New York City and even ships in the Atlantic saw an accumulation of Kansas dust. So human well-being was obviously affected, being connected to the ecological system. 80% of folks required government assistance, and 25% of people moved after Black Sunday. The suitcase farmers, kind of the weekenders, they just really, they didn't really have any ties to the land, as I mentioned earlier. They didn't have any commitments. They just abandoned the land. So lands are just abandoned and exposed. Major health effects, dust pneumonia. 1% of children actually died. Suicide rates went up. And another sort of ecological factor associated with the loss of diversity was a locust plague. This is a picture in California. They actually had to break out flamethrowers to get rid of all these locusts. You should check out Ken Burns' documentary. It's 4 hours long, but it's really interesting. That's where I obtained most of this info from. I got to watch movies to prepare for my talk. So what did management do? Well, they remained committed. They almost gave up. They questioned, should we give up on the West? And eventually people said, well, I don't know. If this erosion creeps eastward, we're in real trouble. So they decided to fix the problem, establish some New Deal programs, established a Soil Conservation Service, which to this day still exists, but it's called the NRCS. Established soil erosion as a national menace. Realized that 80% of the plains were eroding. And perhaps most important, they acknowledged that humans were at fault for this. So the Government Soil Conservation Domestic Allotment Act, they enacted that, and that gave grants at the federal level for demonstrations of new technology. But perhaps another really, perhaps the most important thing is empowering locals through the creation of soil conservation districts. This gave people or farmers the ability to actually seize their neighbors' lands if they weren't practicing proper technique, because all it takes is one farm to ruin the next downwind farm or downstream basin if we're talking about watersheds. So there's a lot of similarities between this, and I'll sort of recap those later on in the talk. So this ended up being the worst man-made environmental problem of all time, and ultimately was a net drain on the economic output of the United States. The government entered agriculture and never left. So we can, some legacies of this new soil conservation techniques, though, are still in practice today, along with some new ones. Planting perimeter trees, shrubs, things like that to provide protection from winds. Restoring the natural biodiversity, using only the right crops for the area. This is an important one with implications for the sediments as well. You want to have earthworms to provide tillage for the soil to allow infiltration of water, and for sediments it's water and oxygen. And finally, you want to have the right balance of minerals and nutrients. And so the National Resource Conservation Service, to this day, they maintain environmental quality incentives programs where they'll actually pay farmers to leave lands fallow for a year. So don't plant any crops on this year, and we'll pay you just because there's an environmental benefit associated with it. There's also a series of national conservation practice standards. These are basically best practices to be followed by farmers. There's, I think, over 200 of them for all sorts of topics. So ecosystem services, there's 4 different types. And 3 are pretty, well, 2 are pretty obvious. Another one is, or the other 2 are kind of behind the scenes. So when you think about what goods you can get out of an ecosystem, the most obvious ones that come to mind are provisioning services. So these are things like food, water, wood, things that really have a direct market value to them. There's cultural services, like recreation, things like that. There are then behind the scenes, more so, are the regulating services. And these play a really big role, although you don't see them and they're hard to quantify. These are things like carbon storage, nutrient storage, water storage, pest suppression. And so those 3 fall over here. And notice they're separate from the supporting services. And the supporting services is more like the glue. So these are the things that are required to maintain this ecosystem intact. And these are the things that you really take for granted, right? But think about if you wanted to replace these services. What if you wanted to grow a forest? What would that take to provide every single piece that comes into growing a forest? These are all accounted for in the supporting services. And of course, all these services together, they all relate to human well-being. So if we take those ecosystem services, all of these things right here, and human health, and we put them in this box right here, and we say, well, this is the condition of the environment, all those things we just talked about. Then this is how we can conceptualize drivers of change. So you have fundamental forces, demographic, economic drivers. These things provide pressures on a system, which provide stressors. These are more specific, like chemical, physical, biological stressors that directly interact with the condition of the ecosystem and human well-being. Now, there's this concept, it's called resilience, and I'll talk about it, I think, on the next slide. But resilience basically affects how the condition of the environment is going to respond to these stressors. So an environment that has higher resilience is going to be able to absorb these stressors more easily. And all along the while, at each one of these levels or steps, you have certain indicators that you can actually pull out and measure. And so what is resilience? It's a really popular buzzword right now in our field. It seems like every grant proposal that comes out, or every proposal application package that comes out, probably half of them have the word resilience in the title. You can see in Google Trends, it's really peaked up. I don't know what happened in the last couple of years. I don't know if that's real or not. But like I said, it's the ability of a system to absorb stressors. And so, for instance, take the soil system. You have the condition of the soil. And let's say a nice biodiverse natural soil, the Great Plains in their natural state, are going to be very resilient. So if you just have a normal stressor, a little bit of wind, a little bit of rain, you're not going to see any erosion, no dust, nothing like that. If you have a really bad storm, a really bad stressor that comes around, you might see moderate erosion. Now, of course, you shift over, you plant just wheat, no biodiversity, you have a less resilient system. It's only going to take a little bit of a storm to erode your system, and a more serious storm can do some major damage, like cause the Dust Bowl. And so where I'm going with this is I'm basically saying that sediments provide an element of resistance or of resilience to marine environments, and beyond that, a specific component of sediments, iron oxides, are the things that are really doing the trick there. So you're not supposed to be able to see this, don't worry, but what this is are all the different biomes on the planet, like open ocean, coastal, and then these are all the ecosystem services. And this is a value. I think it's per acre or per hectare or something like that. And so you can actually assign a total value of all of the ecosystem services on Earth. And this is from a landmark paper, I think back in 1994 or 1997, and it has something like 16,000 citations. And the total value is about $33 trillion. And if you look, the most valuable ecosystems are those that are coastal or rainforest. Natural systems are the most valuable. So as you destroy a system, decrease its biodiversity, it decreases in value. Take a grassland, diverse to begin with. By the end of it, you have a farm, an unhealthy farm, and you actually have to end up putting money into it in order for it to generate value. Same thing if you take an intact wetland, it's worth a certain amount of money, but now if you exploit it and use it for intensive farming, you really only extract about half the ecosystem resources from it. And so now going back to that big chart, you'll have to take my word for it, but the wetlands are the ones that have, are the biomes that have the absolute highest value of all the other systems compared to any terrestrial systems or anything like that. And beyond that, the primary value is actually associated with things like nutrient cycling. So the best ecological service that we can get off of the planet Earth is the nutrient cycling associated with wetlands. You know, things like food production, they're less than an order of magnitude of what we get from nutrient cycling. And what I'm going to argue is that sediments are responsible for a large portion of this value, if not the vast majority. But because they're mostly supporting services, things going on in the background like the glue, people don't appreciate them. We don't appreciate the services that sediments provide until they go bad. I'll come back, I'll swing back around to that, but just looking at the major ecosystem threats to our waterways. So if you look at inland water and coastal water, you see that pollution, nitrogen phosphorus pollution, aka eutrophication, is the major one. You go to a marine environment, it's more like exploitation over fishing. Nutrient loading, if you look at the increase in nitrogen transport coming from, well, I guess just out to the river mouth, you will see that the Mississippi watershed has increased by a factor of 2 to 4 times since humans began messing with the system. It's even worse over in Europe. What's really interesting is when you look over time, and you look right now, the total human input of nitrogen is on par with the natural range of input of nitrogen to these systems. So nitrogen naturally becomes available in these systems when organisms fix nitrogen gas, which comprises 78% of our atmosphere. They fix that into bioavailable nitrogen. So the amount of this process that's happening, humans have actually equaled that with regards to our artificial nitrogen inputs. That's pretty frightening when you think about it. So what does eutrophication cause? In a system with no eutrophication, you will have a diverse phytoplankton and plankton community. You'll have a diverse fishing community. You'll have oxygenated sediments with a nice, diverse benthic community. In a eutrophied system, you have a lot of organic carbon, a lot of algae, things like that are all sedimenting out, and they are causing the sediment to turn anoxic. They're consuming all of the oxygen, and not only that, they're also generating sulfide gas, which is toxic, and that can flux out. You decrease your overall diversity in this case. You really promote things like harmful algae blooms. And this was one of my, I laughed, I think, harder at this than I have in a long time when I saw this it's probably a decade ago. There was a study that came out that The Onion satirized this paper, and they showed that, sure enough, you're not going to be able to go to Red Lobster and get an all-you-can-eat crabs. You're going to be stuck eating krill. That's because that's all that's going to remain in 2050. So how many of you have heard of the dead zone? This isn't a joke this time. Okay, so a lot of people. So the dead zone, nutrients come out of the Mississippi River. You have phytoplankton blooms, algae blooms here in the surface ocean, in this coastal area, and it all sinks. It's decomposed in the sediments. It draws the oxygen out of the sediments, and your bottom waters, like the bottom meter or two, end up anoxic. And all the benthic organisms, the crabs, the worms, everything either dies or has to move on. And so there's really very little that's actually alive, and this happens every year. More on a global scale, you have in the open ocean, a lot of times this is a pretty typical profile, you'll have high oxygen right at the surface because plankton or phytoplankton are photosynthesizing, and then they die and they get degraded, and organisms eat them, and then they breathe CO2, but they use oxygen just like we do, and so you get an oxygen minimum zone. There's areas, upwelling areas, where you have natural eutrophication of nitrogen and phosphorus coming from depth, and those areas you will actually have a true oxygen minimum zone, a hypoxic zone in the open ocean. Now, on the far end of the spectrum, areas that are extremely anthropogenically modified, like the Black Sea and the Baltic Sea. The Black Sea has actually been anoxic for over 100 years now. There's been no oxygen below a certain depth, and to make things even worse, you have sulfide that's diffusing up from the sediments and actually being generated in the bottom waters. So this whole bottom area is extremely toxic to most forms of life. Of course, wherever you go, there's always going to be some form of life that can capitalize on these sulfide concentrations, and that is the case here, of course. But this is sort of the general direction. It's like every year, more and more systems tend to move to the right. It's called global eutrophication. So the major questions that I want to know is, what are the ecosystem services provided by marine sediments? And I don't mean quantity, I'm talking about quality of marine sediments. How do we measure and assign them a value? What are the threats to these sediment ecosystems? What properties enhance the sediment resilience and the resilience of the communities that are connected to them? And how do we measure these buffers? And overall, how do we conserve sediment health? Okay, so I could have put this as my first slide, but I figured it was pretty obvious what soils versus sediments were. So anyways, in case you were wondering what sediments are, they're like soils, but they're transported to the coast. They're underwater. Its original natural material was broken down by weathering and erosion of rocks or soils, and they end up reflecting, to some extent, their parent material, although they are altered in place over timescales from, I don't know, a day to hundreds of millions of years until they are eventually subducted and become part of the mantle of the Earth again. I'm only interested in the first 30 centimeters of sediments, though. It's called early diagenesis. Diagenesis meaning what happens to sediments. That's the definition of diagenesis, is what happens to sediments as they age. And I really only care about the surface because that is what governs the fluxes or the exchanges between the water column and sediments themselves. So just everybody knows all about sediment quantity issues, right? They just had a beach renourishment project in Fort Pierce last year. They want to keep the, this is the Army Corps of Engineers that are doing this. They want to make sure that the beach doesn't, or that the Fort Pierce inlet area doesn't disappear. What you don't hear about is the quality of these sediments, right? They're just moving sediments around, but they don't really ever consider, like, are we moving the right type of sediments around these various places? We know that desirable soils are fertile. We want aerated soils. We want soils that are not waterlogged, unless we're trying to grow rice. Rice plants actually do like to have anaerobic waterlogged soils. But we don't really have the same holistic knowledge for sediments. When I say holistic, I mean we know individual biogeochemical processes that can transform and act in sediments, but we don't know the larger, an overall picture of what good, healthy sediment looks like. Marine sediments, this is their composition. Perhaps in order of abundance, but it really depends where you are locally. Oh, I didn't know I did that. Okay. So what is really important, more so, and because it's so variable regionally, we sort of have to look at the Earth's crust overall if we're going to try and say what are the chemical compositions of our sediments. And so you have oxygen, silicon, aluminum, and iron. So iron is the fourth most abundant element in the Earth's crust, but it's the most abundant redox active solid. And that's really important because life has evolved to use it for all sorts of purposes. The currency of life is charge. Right? We need to move electrons around. That's how batteries work. That's how life works. So life has really evolved to use iron. Take a red blood cell, for instance. It has an iron core. So as far as roles for healthy sediments, I stole this chart from the National Academies, and I added a few boxes of my own. A lot of these things are pretty obvious. You know, they protect us from storms. But the carbon cycling, the nutrient cycling aspects, these are the ones that, again, we tend to take for granted. This is I need to explain primary production in order to explain eutrophication. And so what happens is you have nitrogen and phosphorus inputs. They come over land or they come through groundwater, over land, through runoff or through rivers. These inorganic nutrients, ammonium, phosphate, you can add a few protons to it. It's still essentially the same molecule. It just depends on the pH. You add some carbon dioxide to it, and you add some water, hit it with some sunlight, and now organisms have incorporated all of these parameters into a molecule of organic carbon. It's really organic carbon and nitrogen and phosphorus, but for the sake of brevity, I'm just going to call it organic carbon. So this organic carbon, and in doing this, of course, they also create oxygen as well. And so this then sediments at some point to the seafloor, and it's degraded. So the same reaction actually happens in reverse. We end up taking this molecule of organic carbon and remineralizing it. This is an important term in my field, remineralize. It means we're essentially turning this organic carbon back into its elemental mineral forms. So notice we have the same ammonium and phosphate and CO2. The same things that went into making it are eventually going to come back out of the sediments. So you have just this constant cycle here going on. And under normal circumstances, it's not necessarily a bad thing. This is just how life works. So one thing I want to throw into the mix that people don't really think about all that much is iron hydroxide minerals. So now we have iron hydroxides also flowing in from rivers, and they're just going to be sedimenting alongside this organic carbon. So now we have both organic carbon and iron oxides sedimenting together. So now, what do unhealthy sediments do? You guys have probably heard about these with the algae blooms. Sediment health affects those basic glue services, right? The supporting and the regulating ecosystem services. And we really, really observe when the system goes bad. You can promote phosphate and ammonium sediment fluxes, actually from the sediments and into the overlying water. You can promote respiration. You can promote hypoxia, like the dead zone. You can promote toxic sulfide production. We've already talked about these. You could potentially seed harmful algae blooms. You can generate acid, intensify ocean acidification. You could decrease the water clarity. So when these iron oxides here actually dissolve, anything that's stuck onto them can be released into the sediment pore waters and can also flux out. Also, unhealthy sediments aren't going to have seagrass to really, just like the buffalo grass did with soils, the seagrass isn't sort of stapling down the sediments. So having healthy sediments could really affect water clarity. And finally, sediments can release any contaminants or heavy metals that are actually absorbed onto the particulate materials. When the particulates are dissolved during diagenesis, this stuff can be released to the water column, to the sediment pore waters, and flux out as well and pollute the surface waters. And so how does sediment iron hydroxides ameliorate or fix these processes? Most of them are actually mediated by microbes, but essentially what they're really doing is they're moderating healthy redox conditions. The iron oxides are serving as an electron buffer. In general, with these systems, with the sediment system, an excess of negative charge is bad, right? It's like the opposite of oxygen. Oxygen is positive, electrons are negative. Oxygen is good, negative is bad. Negative is more associated with things like sulfide. So the iron oxides are like a sort of a suboxic, a so-so, somewhat oxidized intermediate, but they're more oxidized than the next most oxidized thing in sediments, which is sulfate, the precursor to sulfide. And I know that was a mouthful. So the other thing that, well, I guess this is sort of associated with the first thing, the iron oxides can absorb, they can react with toxic sulfide to form iron sulfide minerals. So any sulfide that's diffusing up from the deep sediments will react with the iron oxides to form these iron sulfide minerals, which I have here in gray. We're going to talk more about this in a bit. Iron oxides also soak up phosphate, and they can neutralize ammonium. So I'm not going to get too much into those processes just for the sake of time, but again, you're going to have to take my word for it. Okay, so I spent a lot of time putting this beautiful chart together, and I think I'm going to use it in every proposal and paper I write for the rest of my career. And, so in a nice, healthy, oxidized system where you don't have a huge amount of nutrient inputs, you just have a nice balance of nutrients coming in. This is supposed to be organic carbon and nutrients right here. You have, your system tends to have oxygenated overlying surface waters. Your system has a nice big amount of iron hydroxides, which as you go deeper in the sediments become more electron dense, and so you actually start to dissolve your iron oxides. So they disappear as you go on to depth and things get more reducing, or again, electron dense. Your oxygen, of course, decreases right at the surface of the sediments. This is usually the case. It's pretty rare that you'll have a coastal sediment in which oxygen can penetrate more than a couple of centimeters. So if you have intense, or I shouldn't say intense, but if you do have, well, you are going to have anaerobic respiration. When you run out of oxygen, the organisms are going to have to do something. They're going to have to use something as a terminal electron acceptor. So they actually convert sulfate to sulfide. So you will see sulfide accumulating deeper in the sediment column. But you don't, and this is the stuff that's actually toxic to organisms. This is the free dissolved sulfide. This, as it diffuses upwards, it's going to react with the iron hydroxides, and it's going to be removed from solution. So you really don't get any sulfide concentrations diffusing up high. Now this is really, really powerful because it means that you can have, and by the way, this is all sort of a hypothesis to some extent. There's a lot of supporting evidence for it. You can have deeper rooted seagrass because they're away from this zone of toxic sulfide. So you can have these nice, big, lush thalassia beds that are thriving because they have this iron oxide buffer to protect them from upward diffusing sulfide. The other thing to note is that your nutrients, phosphate or ammonium, which are generated at depth as a product of anaerobic respiration, they also diffuse up, but when they hit this iron oxide buffer or barrier, whatever you want to call it, they're also neutralized, although the process is different for both of them. Okay, so moving on to a system that's more like the Indian River Lagoon. You have oxygen. Notice your concentration of oxygen is a little bit less than it was over here. And your iron oxide buffer doesn't extend down as deep. So what you have, you don't really have these nice big lush thalassia beds. You can only grow these smaller, these shorter root plants like halodouli. I hope I pronounced that right, Dennis. And one thing I forgot to mention over here is that you have a nice community as well of benthic organisms. And these are just like earthworms. They are irrigating the sediment and allowing this oxygen to penetrate into sediments. So they're a benefit. You want a nice, robust, biodiverse community here. Now, your community starts to die off here. They may get a little poison from sulfide, or they may periodically run out of oxygen. You start to develop harmful algae blooms as you lose the other control, the organisms that may, and other zooplankton that may, for instance, control these harmful algae blooms. Your nutrients also begin to come up a bit further into the sediment column. They're not quite leaving the sediment column. You don't have a large nutrient flux yet until we get to this third system. This is more like the Mississippi River dead zone, right, where you have no oxygen in your bottom water. You have sulfide that diffuses upwards, and there's virtually no iron buffer there. So your sulfide makes it pretty much all the way to the surface. Your nutrients, on the other hand, do reach the surface, and then they also flux out. So you're really sort of eutrophying. Your sediments are eutrophying the overlying water column. Notice your water clarity here starts to get a little bit darker for several reasons, not just sediment fluxes, but also because you have harmful algae blooms. And then finally, in the last system, your sulfide can also flux out. So you have both a high nutrient and a high sulfide flux. And this is when you have a lot of external organic carbon inputs, which are fueling this large respiration process. Okay, I think that might be as complicated as it gets in this talk. So I hope you were able to follow that. I don't think I can make it any more simple. I've spent a lot of time trying to do that. It's just a tough concept. So in the IRL, what happened? So I'm borrowing this figure from a PI here at Harbor Branch, Dr. Mengshun Zhang. He just led a monumental proposal to the NOAA Ecology of Harmful Algae Blooms program. It was due in March 4th, so we were all scrambling, all the faculty here were scrambling to get this done. So in 2011, you had a super bloom of I think it was a picocyanobacteria, I think really started things off. And then, a nanoeukaryote, that's what it was, okay. And ever since then, your ecosystem has apparently been impaired. You've had a constant bloom of Oriambra up in the Banana River. It hasn't gone away. And sort of the rationale, the traditional thinking is that you had a harmful algae bloom. It came, it blocked out the sun, so the seagrass couldn't photosynthesize anymore. The seagrass then died. The harmful algae died, so you had a massive pulse of organic carbon to the sediments. This massive pulse of organic carbon led to sulfidization of the sediments and perhaps has led to any sort of recolonization from the sediments. But there's something that is really noteworthy, especially from my perspective, that happened. After the superbloom, you tend to have a lot more phosphorus in the water column, especially when you look at it relative to nitrogen. And what I think happened, or at least certainly a worthy thing to investigate, and this was a large part of this proposal, is to look to see if perhaps we had permanently exhausted the iron buffer. You have an iron buffer that keeps the sulfide in check. It keeps the nutrients in check from being able to flux to the overlying water. And it's possible that we could have permanently dissolved this buffer. And because this system is so insulated, there's not much irrigation, there's not much exchange with the ocean and things like that, it's really hard to reoxidize and regenerate that iron buffer. So now you just have nutrients that are sort of a free-for-all, just floating between the water column and the sediments, and it could be maintaining these algae blooms. So that's a cool idea that we've been working on. So where's the iron come from? Well, these systems in the southern United States, southeastern United States, Georgia, Florida, there's a lot of iron here. It comes from the drainage basin. So here's the Caloosahatchee River. This is the western drainage of Lake Okeechobee. And you have so much organic matter in the peat, right? It's the tannins, the brown stuff coming out of the rivers here. It actually leaches iron out of the soil. So you end up having a lot of dissolved iron in the river water, but that river, while that dissolved iron is stable in fresh water, when it actually hits salt water, it flocculates, it aggregates, it precipitates. And so it all accumulates right where it encounters salinity, so very low salinities like 2 or 5, and seawater is 35. And so sure enough, if you look right here, the amount of iron hydroxides in your surface sediments peaks right where the river opens up. And we actually saw the same thing in the St. Lucie estuary and in the Satilla River in South Georgia. So we're pretty sure that it's universal to all black water rivers, all organic rich rivers. So we have a lot of iron in the sediments around here. So if we're going to see this effect from an iron buffer anywhere, here is as good of a place as any. Now, this is a timeline that I spent a lot of time putting together, and I think it's going to make for a nice review paper, perhaps. And it's sort of an evolution of the buffer roles, all the different ecosystem services that iron oxides can provide. And I'm just going to talk about a couple of them. Obviously, the sulfide sequestration, but also maybe I'll skip that one just because I don't have too much time. But iron oxides do mediate organic contaminants via an abiotic reaction. But yeah, I think I'm running long, so I'll skip that. So this is some work that Andrew, my student, has been working on. Seagrass is in a global decline. Typically, when people want to restore seagrass, they try and fix water clarity. They try and do that by, well, improving light levels. They try and do that by controlling nutrients, controlling flushing. These are the primary factors. People very rarely actually consider sediment composition. We know that sulfide is toxic to seagrass. If you have a seagrass bed right here, here's the perimeter of a seagrass bed at 0. So if you go into the seagrass bed by a few meters, your pore water sulfide concentrations are low. Outside of the bed, where you actually have dead seagrass on the ground, you have high sulfide. This was a landmark paper from Florida Bay in 1994. Now, a more recent paper has actually showed that for areas that are receiving iron inputs, you have a improved seagrass growth rate. And there was some other work that demonstrated that sulfide is the likely reason for this. Iron can titrate the sulfide and keep it at bay. Now, one thing to note is that this work was done in carbonate sediments. These are white, sand, pristine sediments that don't really have much iron to begin with, not like the IRL. So why should iron be an issue in the IRL? Well, because we have a ton of organic carbon, which remember is kind of like the enemy of iron. Organic carbon is rich in electrons. Iron oxides has a deficit of electrons. But so this organic carbon could potentially be producing sulfide. So how were we going to test this hypothesis? Well, Dennis was nice enough to take us out on to his 4 existing long-term transect sites that I think he's been maintaining for a couple of decades. We sampled sediments using we take a syringe and we cut the bottom off. And we take miniature sediment cores. It's very low tech, but it actually works really well. We analyze the iron content just using an ascorbate extraction. This is just vitamin C. We take a little bit of sediment, we weigh it, we mix it with some vitamin C, and we look at how much iron comes off after 24 hours. And then we could normalize that iron back to the original amount of dry solid. We measure organic carbon using loss on ignition. You essentially weigh your sample before, and then you burn it at 500 degrees Celsius, and you weigh it afterwards. And anything that's lost is organic material, and you can use a conversion factor and derive your organic carbon from that. Seagrass species and percent cover, that's sort of our dependent variable. Dennis's student, Dennis's technician, John Hart, would actually go out there and snorkel in 1 foot of water. It's kind of ridiculous, but it's effective. He didn't want to go out on the cold days, though, so I think we missed a sampling in December. But so what we're really trying to do is come up with a picture of what the ideal sediment conditions are. And this is funded by the Harbor Branch Foundation through the state license plate. This was a seed project that Dennis and I had. So our preliminary results, I don't know if they're so preliminary anymore. I should probably start saying these are our actual results. We do, in fact, see an effect. If we look at percent cover, so a healthy seagrass system is going to be yellow. We see that in impaired system where you have very low, where you have a low seagrass cover is going to be in purple. we see that you have more impaired sites at higher ratios or higher concentrations of organic carbon relative to iron hydroxides. So we shouldn't necessarily have two boxes. Just because we find an impaired growth up here in this box doesn't mean that you have sulfide poisoning. There's lots of other reasons why you would expect to not be able to grow seagrass. But this area right here does seem to suggest that organic carbon inputs or organic carbon excesses are having an effect. So what we can sort of boil this down to is we can say that sediments that have organic carbon to iron hydroxide molar ratios of greater than 200 may have a reduced chance of survival. So now people can go out here, sample wherever they want to do seagrass restoration, right? If you're going to do a transplant, if you're going to make a decision to stop Lake Okeechobee discharges because you want the water to clear up so that you'll have a better chance of growing seagrass down in Stewart. Well, if the sediments aren't going to support it, then why bother doing it? So now you can test sediments to make sure that they won't be subject to sulfide poisoning. So pretty cool, positive finding. So this is a cool technique that our lab has, and I'm just going to breeze through it. We use these glass electrodes. We pull the glass, and they're only they're less than a millimeter in diameter here at the tip. If you zoom in, there's a 100 micron diameter. That's 0.1 of a millimeter. 100 micron diameter gold wire that we amalgamate in mercury, elemental mercury. And then we can actually take this electrode and penetrate the sediment. This is a sediment core. We can penetrate it in very small increments. It takes a few hours. But at each depth, we stop and we run a series of voltammograms. This is an electrochemical technique. And so we can measure all these various things that are highlighted in red. We can measure all these analytes using a few different types of scans. Notice sulfide, iron, oxygen. So it's a really powerful technique for painting a nice illustration of what is going on in sediments with respect to oxygen, iron, and sulfur. Lots of other things. I won't talk about those. So this was perhaps the single greatest month of my life. I think I can honestly say that the most fun I've ever had in my entire life was spent on this research expedition back in 2012. We were in the Congo River deep sea fan. So the Congo River is one of the only ones that is still connected to its deep sea fan. So you have these gigantic, massive turbidite flows, these underwater plumes of sediment that come off of Africa, which is, by the way, the red continent, right, because it's iron rich. So you have a lot of iron oxides that are funneled all the way down and into the deep ocean, 5,000 meters depth and 800 kilometers offshore. So it's like having a coastal ecosystem that's been transported way out to the ocean at depth. So it's a really cool thought experiment. It's like a natural experiment in a way. We had we crossed the equator, so we of course had a hazing ceremony since that was my first time crossing the equator. I guess it's legal in international waters or in France, I'm not sure. But we would collect sediment cores using a multi-core, and this is a really interesting contraption. It would take about 3 hours to go down, take a sediment core, and 3 hours to come back up. We also had an ROV. You can see it being deployed there. The ROV could take smaller sediment cores from very precise areas. Obviously, when we're dropping this thing off the back of the boat, we don't know where it's going to end up. So when we were out there, right, it's just like a blanket of brown material. It's iron-rich, muddy, clay, fine-grained sediment. But every so often we would find these black patches, let's say like, I don't know, maybe one every 10 acres or so, and they're about the size of a desk. And you have all of these clams around the edges. They're called vesicomyidae. And what you notice is you have clams around the edges, but you don't have clams right here in the middle. So we wanted to compare these sites. We wanted a core from right here to look at the geochemistry, and we wanted a core from where the clams were. So I'm going to compare these two sediments. So using this voltammetry electrochemistry tool that I described on the other previous slide, you can see as we profile the sediment and we go down, here's where we hit the surface. You can see that sulfide here in this dark yellow line. It increases. It's right at the surface. So sulfide is right there. And that's why the sediment is black. And it reaches very high concentrations, greater than 2 millimolar. If we look at where we had a clam, on the other hand, you don't have sulfide, look, until 4 centimeters down. What do you have? You have dissolved iron. So it's like you have a zone of dissolved iron cycling here, and somehow that's keeping the sulfide at bay so that the clam can survive. Otherwise, this area right here where there's sulfide all the way up to the surface is too toxic for the clam to survive. And so, sure enough, we didn't need to do all this complicated experiments to tell us that, but just by looking at the core, you could tell it's very sulfidic. Right here, you have this black iron sulfide sediment, and here is your nice brown iron oxide-rich sediment. And this was actually where we got the original idea for iron oxides being a buffer. It's like iron oxides were creating a habitable zone for these clams. It's really, really significant. Now, I've convinced some collaborators here at Harbor Branch to trust me and let us actually go out and do this type of work looking at clams in conch now. So I'm going to be working, or we have proposals in the works to work with Dr. Megan Davis, Matt Ajamian, and Susan Larimore. I said I wasn't going to talk about that. Okay, so now I'm just going to tie it all up and tell you about my vision for what sediment health conservation might look like. We realized all too late from the Dust Bowl we were making huge mistakes. There's a lot of similarities between the two, whether you want to look at just the level of soils and sediments themselves, regionally, like the Great Plains versus the IRL, and just globally. So what can we learn by comparing these systems? And I had like 3 tables with 50 notes. I was taking active notes when I was watching that documentary. Happy to discuss more after the talk, but I'm just going to point out the ones that are bold. These are some analogies, right? In the Dust Bowl, non-committal, people didn't care if they lost their land. They invested like $6 or something to get their loan. They didn't really care. So no stewardship. Same thing in the lagoon. The problem creators aren't directly tied to the lagoon. Natural human pressure, stressors, and health indicators. We are clearing windbreaks. Or they cleared windbreaks back in the day, giving the winds access to the soils, therefore creating the dust bowls. These days, we are clearing mangroves and installing seawalls. We have no natural filter for contaminants. Also, mangroves buffer against storms as well, but that's not so much related to the sediments. And finally, the perception. This one's interesting. It was good earth when it was growing their crops. It was dirt when it was in their noses. Now it's muck when it's fluxing nutrients and causing harmful algae blooms. And now I'm trying to say it can also be a resilient buffer. Eventually they adopted responsible farming methods. They restored earthworm aeration. They restored biodiversity and the really important keystone species. I actually submitted a proposal to the Golf Research Program with a faculty member here who spoke about a month ago, Dr. Tracy Mincer. And we had talked about making an amendment. So now you can amend sediment. Instead of just trying to, let's say, transplant seagrass, you have to transplant seagrass with minerals like iron oxides with microbes. You got to have all 3 facets of the ecosystem if you want to succeed in restoration. It wasn't encouraged. It was a letter of intent, but it wasn't encouraged to submit a full proposal but they had legitimate concerns. So the first thing that I think we want to do, I'm going to talk about three things. This is the first one, is we really want to get to know the soils or the sediment system. We want to get to know it independent of everything else. People study wetlands. They study seagrass. They study the system as a whole, but people don't really study the sediments all that much. And I know it's kind of opposite, right? Normally we want to study everything in a holistic sense. But in this case, it's important to study sediments on their own. We want to implement routine sediment monitoring, or we want to augment existing monitoring programs like those that are going on for seagrass or harmful algae blooms. We want to define what is sediment quality, what is sediment resilience, and how do sediments get that way? How is that naturally partitioned? So the Indian River Lagoon National Estuary Program just revealed this, I think, at the Indian River Lagoon Symposium last month for the first time. They came out with this vital signs, and it's really cool. I think it's 32 individual parameters that need to be considered or monitored for us to create a healthy lagoon. And what I see is legacy loads. They're talking about muck there. That's legacy loads. There's no other reference for sediments. Legacy loads falls under water quality. Maybe habitats, you have wetlands. But sediments themselves don't have anything. Me being a, remember, you have to remember that these, the whole point of my talk is that non-muck sediments, like sandy sediments, muck is only 10% of the lagoon floor. The other 90% is regular sandy sediments that provide all sorts of ecosystem services. So I would argue that sediment quality should have its own group, but of course I'm biased. It's probably too much, but I don't know. But I think certainly sediment biogeochemical resilience or something of that sort should have its own little wedge, though. This was a missed opportunity, though, because last year the IRL asked for feedback on this and didn't really put this idea together then. So I didn't submit anything. I was also really busy working on other stuff, but that happens. So, okay, if we wanted to quantify biogeochemical resilience, how would we do it? And this is one of these situations when you think you have such a really cool original idea, and then you go in the literature and somebody's already done it. And you say, oh, no. But really, this is the only paper. It was a 2004 paper that is the only paper that I found that actually looked at something like this. And they weren't looking at resilience. They were looking at the opposite. They were looking at vulnerability. They wanted to do it for the purpose of analysis, comparison, and classification of sediments and their overlying water bodies. I think we can use it to look at tipping points of systems. We can prioritize areas for conservation, and most importantly, assign a value so that we can get a market going on for these things. So they looked at similar concepts. They agreed that higher flushing is good, lower carbon inputs are good, higher iron oxides are good. They also said that higher carbonate minerals are good, something I didn't think of. Carbonate minerals capture phosphate and they can buffer acidity, so it's something we should look at. So I just did a little experiment where I took an Indian River Lagoon sand and I used their system and I assigned a ranking, and the Indian River Lagoon has a potential vulnerability of 9 out of a total possible 18. So we're like halfway there. The regular Indian Lagoon sediments are pretty vulnerable, but they're not the most vulnerable sediments in the world. I think the opposite. So of course I think positive, right? So I'm thinking about resilience instead of vulnerability. There's a lot of things that I would improve about this, about their procedure to quantify vulnerability. I would actually try and relate these parameters to each other, because right now you're essentially just categorizing things like, for instance, reactive iron. You look at the concentration. If it's greater than 200, then you get 3 points. 3 points for reactive iron. So it's just kind of like, you know, it's kind of arbitrary. Sure, things you rank them, but then you end up just assigning an arbitrary number. So I want to actually use geochemistry and develop relationships like we did for the seagrass when we said that molar ratios over 200 are going to prohibit seagrass growth. just details about how I extract iron. What else? Oh yeah, we want to incorporate some newly discovered roles. So since they came out with that paper, we've learned more about what iron oxides actually do in systems. And the other thing to incorporate is the microbial community structure, again, because most of the processes are mediated by microbes. The second thing we want to do is incentivize, aka money talks. We want to assign a value to our ecosystem services that sediments perform. And we want to potentially implement a payment for ecosystem services. This is a new idea that's catching on. And an example for the IRL would be like, well, let's pay people to not destroy their natural mangroves on their property, and instead and replace them with a seawall. Because I care so much about the lagoon, I'm going to agree to pay these people a certain amount of money to keep their land natural because it protects the ecosystem services associated with my lagoon. But I think a cool idea, and this is something that I came up with, it might be just crazy, I don't know, but take advantage of aquaculture. So what we learned from the Dust Bowl is that you got to have ownership. You got to have people speaking for the sediments, people that have a vested interest in the sediments. So what if we had sea vegetables growing in sediments in the lagoon? And of course, now people would have, if we gave out like leases, like aquaculture leases, long-term, 10 years, let's say. And now folks that are growing the folks actually doing the aquaculture are certainly going to try and protect the lagoon. And they'll make sure it stays safe locally. But it's also going to give the lagoon a voice. Because these people care about their crop, they're going to fight for the sediment health when they go to city council meetings, when they talk to their politicians. So they could, potentially stop Okeechobee discharges or pollution coming from agriculture, things like that. By having folks that have a vested interest in the sediments, we give the sediments a voice. And I have a pending proposal as well with the whole Harbor Branch integrated multi-trophic aquaculture system. There's about a dozen PIs here, principal investigators here at Harbor Branch who are working on this, and I hope to be working on optimizing sediment conditions for growing sea vegetables because we know that they grow better in sediments than they do just in water or not sediments. 3, last thing, we want to be able to rank the quality and the resilience so we can prioritize restoration. We want to be able to actually simulate restoration effectiveness with a diagenetic model. So here you have folks, they go out and they map muck in the IRL, and then people go out and they dredge the muck. It's something like $200 million that Brevard County is going to be putting up to dredge muck over the next decade. Instead of going out there and actually just pulling out the muck, what I think we should do is instead conduct simulation experiments. So we can model the sediment system. This is called a diagenetic model. These points are individual measurements, and then we actually understand the system so well that we can reproduce it based on purely fundamental reactions, and that's what these lines are. So we can do experiments, simulation experiments, where we, let's say, remove a large pulse of organics from the surface sediments, and we could look at how the ammonium flux responds. And we can do this before we go out and do something like dredging, and this could potentially save us a lot of money in the long run. So it's a little bit of an investment with a potential long-term payout. I already mentioned this probiotic idea, so we could actually go out there and try and increase the resilience of these systems. Last but not least, it's important to educate and empower the committed stakeholders. We want to lobby for sediment conservation to actually have sediment conservation agencies, and whether this just means that sediments become a more important part of the water management districts, that's okay too. And we need specific laws about protecting sediments. And finally, we need a set of conservation practice standards, just like they did, best practice standards to deal with sediments as we do with soils. The Indian River Lagoon, we're in an ideal site to explore these. We're specifically at Harbor Branch, we're obviously close to it. It's very biodiverse, lots of different habitats. It's insulated, lots of contaminated inputs. Sediments are vulnerable here. We're susceptible to climate change, sea level rise, global warming. Our communities are particularly affected by the harmful algae blooms, so there's certainly justification to it. The folks at Florida Institute of Technology have already opened the door for sediments, so we just can kind of scoot on in there and expand it some and sort of change the paradigm that mud isn't necessarily a bad thing, right? It could be good too. And we should augment existing monitoring programs. At Harbor Branch, we have all the expertise needed to do all this sediment work that I've just described. I'll just give you a second to glance over that. But where do I fall? I'm sediment health diagnosis and monitoring tools. Okay, so I'm done talking about sediments. Now I just want to touch on a couple other projects before I turn away. I've already talked about these sediment projects with respect to enhancing the resilience of sediments. We're also currently working on developing a benthic lander that can be deployed in blue holes in the Gulf of Mexico so we can explore the sediments at the base of them. We're designing biofouling resistant surface coatings, and we're testing them to try and make it so that suckerfish can't stick to autonomous underwater vehicles. Yeah, it's tens of thousands of dollars in damage caused by those suckerfish. You guys may have seen this in the news recently. Just February 5th, this came out. We're working with a small company that makes an autonomous sailboat, and we used it for harmful algae bloom monitoring in Lake Okeechobee. It's totally cool, completely autonomous. You can give it waypoints, or you can actually control its individual, the sails, rudder, things like that. And we sent it along a star-shaped pattern with a repeat visit to this NASA Sea Prism observing platform that's out there. And we're actually going out next Monday and Tuesday. We're going out to Florida Bay to try and see if we could get this sailboat to work down there, because it's such a difficult region to work in because it's so remote and shallow. And of course, I couldn't help but take a mud sample when I was out in the lake. And sure enough, there's a ton of iron out in the lake. So I certainly intend to explore the coupling or the role that sediments may play in causing the microcystis blooms out there on the lake. And so I just wanted to thank everybody at FAU, our collaborators at my old institution, Sanibel Captiva Conservation Foundation, of course our funding agencies, and that's about it. So happy to take some questions.